Hi guys, today we're solving Cambridge A-Level Biology, Paper 1 Topical, starting with Chapter 1. Question number 1. Which definition of the magnification of a drawing of a leaf is correct? So remember, for magnification, this triangle would be helpful. You can remember it as I am. To find the magnification, you do image over actual. So which one would be correct out of the options given to us? The actual size of the leaf multiplied by the magnification. So multiplied is wrong because we divide. The measured difference in size between the leaf and a drawing of the leaf. That is wrong because you don't take the difference. The increase in size of the leaf when observed using a microscope, no. The accurate definition would be this one, because the size of the drawing of the leaf divided by the actual size. That's what's happening here. Question number 2. The electron micrograph shows part of 2 cells. This is one cell and this is another cell. Which labeled feature identified these cells as eukaryotic? Remember, eukaryotic, the one difference between eukaryotic and prokaryotic cell is that eukaryotic cells will have membrane-bound organelles, so that's what we're going to look for here. W, as you can see here, is the chloroplast, and that is a membrane-bound organelle, so that would be our answer. X is cell wall, and Y could be the cytoplasm. So the only membrane-bound organelle here is chloroplast, so that's W only. Question number 3. Plant cells are stained and viewed using a student microscope. Remember, these things are important. It's a student microscope and the light source is a natural light. What could be clearly visible at only 400 magnification times 400. So this order is important to remember. The most obvious one would be nucleus because it's the largest, and then you have chloroplast, you have the mitochondria next, in order of largest to smallest. Then the smallest typically is ribosome at the end. So in this case, they've given us cristae of mitochondria. They're not just saying mitochondria, but cristae of mitochondria, which is going to be much smaller than grana of chloroplast. That's again within the chloroplast. It's going to be very small. Nucleoli, that is the stained part within the nucleus, so that's likely to be the answer. And ribosome is the smallest, so definitely not this and not these. The answer is nucleoli. Next question. Which lengths are equivalent to 1 micrometer? So let's just write down the conversions between unit. Millimeter to micrometer to nanometer. So this is from largest to smallest. The difference is 1,000 between these. And when you go from this large to small, you're going to multiply it. And then you go from small to big, you're going to divide it by 1,000. Now let's see. 1 micrometer is this. 1 micrometer is how many millimeter? That is going to be 1 divided by 1,000. So 1 micrometer is going to be 0.001 millimeter. And then the other way around, 1 micrometer can also be given in nanometers, which is 1 micrometer is, as you can see, you're going to times it by 1,000. You have 1,000 nanometer. So these are the only two answers, this one and this one. Everything else is wrong. You can see why, because 1000 millimeter, that's never the case. Okay, 0.01 nanometer, that's wrong because you always multiply, so it's not going to be like a decimal number. And this is too big. From micrometer to nanometer is just a difference of thousands. This many zeros is not possible. And to get it into millimeter, you have to divide by 1,000. So this looks like it's divided by only 100, so that's not enough. So these two are the only answer. 3 and 6, that is D. Next question is question number 5. Some secretory cells synthesize and release glycoprotein. What is the correct sequence of some of the events that occur in a secretory cell? So the first one they've given is exocytosis. Remember, this is going to be the last one because it's moving out of the cell. That's the last step. It moves out of the cell. So, one has to be in the end. So we're just going to get rid of every other option that doesn't have one at the end. So it's either B or D now. Let's look at the other steps. So mRNA binds to ribosome. This would be the first one. Translation happens. And then you have the synthesis of glycoprotein has to happen. It has to be synthesized. That is 2. Then you have products accumulate in secretory vesicle. Obviously, all of that is going to be packed. And then in the end, it's going to be sent out. So it's 3, 4, 2, 1. 3, 4, 2, 1. That is B. Next question. A scientist used a blender to break open a sample of animal cell and release the organelles. The extracted mixture was spun in a centrifuge at a low speed. The heaviest type of organelle sank to the bottom of the centrifuge tube to form a solid pellet 1. The liquid above pellet 1 was poured into a clean centrifuge tube and spun at a higher speed. The next heaviest organelle sank to the bottom to form solid pellet 2. So you have solid pellet 1 formed, and then the next heaviest is going to be the pellet 2, and then you're going to repeat it twice more to get pellet 3 and pellet 4. So it's going from heaviest to lighter, each containing a single type of organelle. What is the function of the type of organelle extracted in pellet 3? So we need to know what is pellet 3, which is the third heaviest, and then we're going to be able to find out the function from there on. So let's just see what we've got here. Digestion of old organelles, that is lysosome. Production of ATP, that is mitochondria. Synthesis of mRNA, that is in the nucleus. Synthesis of protein, that is in the ribosome. Now that we know what these functions are for, we can also know that heaviest at the bottom is going to be nucleus. And then you would have what's heaviest is nucleus, and then you'd have mitochondria, and then eventually you'd have the lysosome and then the ribosome. Okay, so it's this way. That is towards the lightest. So the third one would be lysosome, and that is A, digestion of old cell organelles. Next question. Which cell structure contains cytoplasm? So chloroplast, not really, because it the inner foldings form the cristae, and then it has the matrix in the center. Mitochondria? No. Plasmodesmata, that's the one that's directly linked to cytoplasm because it joins the two cells in plant cells. It joins the two cells and it's a bridge of cytoplasm. So this is going to be our answer. And it's definitely not the smooth endoplasmic reticulum. Question number 8. Some cell structures are listed in a particular order. You have nucleus, ribosome, Golgi body, and vesicle. What determines the order in which these cell structures are listed? So you need to figure out what this order is for. So you have sequence used in synthesis of a lipid. So synthesis of lipid, by the way, is SER. But you can see we don't have SER, smooth endoplasmic reticulum, given. So that's not going to be our answer. Sequence used in synthesis of an antibody, which is a protein. So you can see, so it's a nucleus, transcription happens, then ribosome, translation happens, Golgi body, the packaging happens, and then vesicle, where it's released in the form of vesicle. So that's going to be our answer. Let's see why not the other ones. Size from largest to smallest, that's not the case because ribosome should be the smallest. And then D, size from smallest to largest, that's not the case because nucleus is not the smallest, it's the largest. Question number 9. The drawing has been made from a section showing part of an alveolus and a red blood cell in a capillary. So you can see that here. The magnification of a drawing is times 5,000. What is the minimum distance that oxygen must diffuse from air? Minimum distance oxygen must diffuse from air in an alveolus into the red blood cell. So you know that the alveolar epithelium has very thin wall, and then the capillary endothelium that is also very thin of the blood vessel, and it's about 0.5 micrometer to 1 micrometer. So let's look at our options here. You have 2 nanometer options. We're just going to get rid of those two because they're too small. So you have either 0.1 micrometer, or you have 0.1 would be too small again, so it's 1 micrometer because you have like multiple layers that it needs to diffuse through. Next question, question number 10. The antibiotic chloramphenicol inhibits protein synthesis in mitochondria and in some prokaryotes. In protein synthesis in mitochondria, chloramphenicol does not inhibit protein synthesis in the cytoplasm of eukaryotic cells. It does inhibit protein synthesis in mitochondria and some prokaryotes, but not inhibit protein synthesis in the cytoplasm of eukaryotic cells. So that's going to be nuclear DNA related. Let's look at it. What would be the effects of the cells of a person being treated with this? The rate of ATP production is going to decrease. Why? Because the protein synthesis is inhibited, so the translation stops in the mitochondria. As a result, the ATP production is also decreasing. You might think that it has no effect on the DNA because of this part, that it does not inhibit protein synthesis in cytoplasm, but it does have an effect in the end because you have less ATP, so you're going to have decreased transcription of nuclear DNA. That is A. Okay, question number 1. This is a common kind of question that has come up a few times. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by a number. So you have these functions. The first step would be to identify what the structure is, and then you're going to match it with its appearance given by these letters. sorry. So the mRNA passes through to the ribosome. That's going to be like nucleus, could be nucleus. Synthesis of polypeptides, that's ribosome, because ribosome is the site of protein synthesis, and packing of hydrolytic enzymes that will remain in the cell. So you might think this is going to be a lysosome when you read hydrolytic enzymes, but this is the key word here: packaging. Packaging is done by the Golgi body. Now, appearance-wise, let's look at this. V is membranes which surround an enclosed inner cavity. That is like SER, RER. That's not any of our options. Non-membrane bound spherical structures. So what is that? That is ribosome. Ribosome is spherical and it's not membrane bound. Double membrane interspersed with pores. What is that? A nucleus. Because nucleus has nuclear envelope, right? Non-membrane bound cylindrical structures. So that's none of our options here. That's more of centriole. When we talk about cylindrical structure, Z is membrane bound sacs arranged as flattened stack. When you talk about flattened stack, that's when you think of Golgi body. So this is one of our options. So let's just like put the correct number next to it. Golgi body is 3. Nucleus is X is 1, and this is 2. So you have 1 being X, 2 is W, and C is our answer. Question number 12. A student calibrated the scale on the eyepiece reticule in the eyepiece lens of a light microscope. The student was given a stage micrometer scale to use. The divisions on the stage micrometer were 0.1 millimeter apart. Which data must the student collect in order to calibrate the eyepiece reticule? So you need to know how to calibrate the eyepiece reticule. There can be questions asked in a lot of ways for this. So which data do we need for this? Let's look at what options they've given. Magnification of the eyepiece lens. No, we don't need magnification. Number of divisions of the stage micrometer scale seen in one field of view of microscope. Yes, we do need that. And number of divisions of the eyepiece reticule scale equivalent to each division of the stage micrometer. We don't really need that data. So it's going to be 2 only. That's C. Question number 13. The diameter of a living cell varies considerably. The diameter of typical eukaryotic cell is this much, and prokaryotic cell is this. Using these measurements, what is the maximum number of each cell types which could fit along a line 1 centimeter long? So what I would do is I would find out what these numbers are. So this one is 1.5 times 10. So you just move the decimal 1 times towards the right. You end up with 15 micrometer. And then in this case, you have 7.5 times 10 to the power 2, which is 100. So you're going to move it 2 times. You're going to end up with 750. And this is nanometer. So don't mix up the units. This question is just testing whether you're able to calculate convert the units back and forth or not. Then I'm gonna put them in the same unit, so I'll just make this nanometer. 15 micrometer. Remember, micrometer to nanometer. When I'm going down from big to small, I'm gonna times it by 1,000. So this is 15,000 nanometer, and this is 750 nanometer. Now, Or since they've given it in centimeter, how about we just keep everything in centimeter? So millimeter, centimeter. So this is times 10 and this is times 1000. So 15 micrometer into micrometer is here, and you're going to make it a centimeter, so you're going to divide it by 1000 times 10. That is 10,000. When you divide 15 by 10,000, you end up with 0.0015 centimeter. And when you divide nanometer, so nanometer to centimeter, that's going to be, you're going to do, there are 3, 6, 7 zeros, or you can do it step by step, but I'll just do it directly. So when you divide it by that many zeros, you end up with 0.0040 zeros, should be there, centimeters. So I have both my numbers in centimeter. Next thing is how many can fit in. So I will divide each by 1. Sorry, divide 1 by each of these values. So I'll have 1 divided by this. That's going to give me around, it should be like 666. And it keeps. And this should give me, again, if I divide 1 by this number, because we need to see how many of these we can fit within 1 centimeter. So it's going to be 13,303.33. So let's look at the options given to us. I just like to convert these back to numbers that are easier to compare. So times 10 to the power 2, that is times 100. That means you move the decimal twice, so it's 1.3 like this, and you end up with 130. In this case, it's going to be another 0 because it's to the power 3. To the power 4, it's just another 0, and 5, it's one more 0. So 4 zeros. Same thing here, 6.7 times 10 is 67, times 10 to the power 2, that is going to be 670. This is going to be 2 zeros and 3 zeros here. Now let's just look at what's the right answer. So for number of white blood cells is this one. We got 666. That is close to 6. If you round it up, it can be 670. So this would be our answer. And in this case, you can see we got 13,000 in 13,000. So that's going to be the 1,000 option, not the 100 option or this one. It's going to be 13,000, which is this. Question number 14. Which structures are found in animal cells and in plant cells? So in both. That is going to be centriole. Remember, centriole is only in animal cells. Lysosome in both, nucleolus in both, because both have nucleus, they're going to have nucleus. Vacuole, so in plant there is large permanent vacuole, but it can also be found sometimes in animal cells. So yes, so it's 2, 3, and 4, that is C. Question number 15. Which features shown in the diagram can be present in eukaryotes? Present in eukaryotes. So you have DNA, RNA, 70S ribosomes, and peptide glycan cell wall. You know, DNA is present in eukaryotes, you know that. RNA is also present because remember, there is mRNA, tRNA, there is RNA. So those are present. And also, you know that how the protein synthesis happens, so there's a lot of RNA involved. 70S ribosomes, so this is where you might think that it's not there, but remember, it is not present in the nucleus or the cytoplasm, but it is present in the mitochondria of the eukaryote cells in animals and in the chloroplast in plant cells. So this is present. Otherwise, it's ATS ribosomes. This peptide glycan cell wall, no, because this is in prokaryotes, bacteria. Otherwise, if there's a cellular cell wall, then you could consider this yes, because in plant cells you have it. Answer is 1, 2, and 3. That is A. Next question. This is similar style question to what we did before. First step is to identify the structures, what organelles they're talking about, and then you're going to match them with their appearance. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by a number. Organizes microtubules to produce the spindle during cell division. That is centriole, or even centrosome you can say, because 2 centrioles make up a centrosome region. Synthesis of polypeptide, that is ribosome, site of protein synthesis. Synthesis of lipid, this is SER. Remember, SER is responsible for the production of lipids and steroids. The appearances were listed by a letter. Now the obvious one first, non-membrane bound spherical structure, you know that is ribosome. Okay. Double membrane interspersed with pores, that is nucleus or nuclear envelope. You're not talking about that here. We don't have that in the organelles that we've identified. Y is non-membrane bound cylindrical structure. You know that is centriole. So we have that. C is, now V and C, this is a common mistake to make. So you might think membrane bound sacs arranged as a flattened sac is SER, but anytime they use flattened sac, that's more of the wording used for Golgi body. And for this, they're going to talk about, in this part, they would talk about packaging or processing of something. And since they talked about site of lipid production, that is SER, and this is going to be the answer for that. Membranes which surround an enclosed inner cavity. So this is SER, because it describes the tubular network of SER, remember? Okay. The one that matches our answers is, let me just put the numbers next to it so it's easy. Centriole, that is 1. Ribosome is 2, and this is 3. So this, and this, that is C is our answer. Next question. Which type of RNA are present in prokaryotic cells and in eukaryotic cells, in both? So all of them are going to be present, so it's A. How many nanometers are there in 1 millimeter? So go back to the conversions. You have millimeter, micrometer, nanometer. So in 1 millimeter, you're going to have 1,000,000. So that's 6 zeros, and this is going to be your answer. Which cell structures contain 70S ribosomes? So 70S is in the chloroplast, in the mitochondria, nucleus has 80 S ribosomes. Cytoplasm also has 80 S ribosomes. So it's 1 and 2. Next question, it says the electron micrograph shows part of a plant cell. So let's identify what these are. P is cell wall. Common mistake is to label this as cell membrane, but it's way past the cell membrane. So this is cell wall, and it's a plant cell, so we know it's gonna have a cell wall. Then you have Q. You can see it's plant cells have large permanent vacuole, so that looks like a vacuole. R is going to be your chloroplast. And S is, you can see this is the nucleus, and within the nucleus is your nucleolus. Which combination correctly identifies the label cell structure? P is cell wall, Q is vacuole, R is chloroplast, and S is nucleolus. So D is our answer. The next question is a little confusing, but as long as you know your conversions and the formula for magnification, you're good to go. The student was told that the actual length of a cell structure is 5 micrometer. Actual length is 5 micrometer. The student was asked to state an equation that can be used to calculate the magnification of an electron micrograph of this cell structure. So it's not going to be the standard one, it's going to be slightly different using what they've given you. The student used some of the letters Q to U in the equation. Some of these have to be used. So let's look at our triangle. Typically, it's going to look like, remember the triangle from the first question? It's I am. So magnification is image over actual length. They've given you, so this is a standard one, so we're going to see how we apply this to fit into our standard formula. So actual length was given as 5 here, and you know that is U, but if I try to, that means U has to go as the denominator, right? But if you look at all the options here, we don't have U for any of them as the denominator. We don't have U. In this case, we have U, but it's multiplying. U is never going to multiply, so you're going to get rid of those answers. But in replacement of U, you have 1 over 5. So that we can use. That way we are going to have 5, which is our U, that is our actual length, as the denominator. So we're going to have it as 1 over 5, which is T. So now you know T is our actual length, and that is 1 over 5. You're just going to have to times it with the image to find out the magnification. Now what's the image? The image of the length of the cell structure image on the micrograph in centimeter, and the same thing in millimeter. So yeah, now you need to know which option you're going to pick. So since our actual length is 5 in micrometer, we need to keep our image length also in micrometer. So centimeter to micrometer conversion and millimeter to micrometer conversion. Remember that centimeter to micrometer, you're going to need 10,000. We don't have that, we just have 1,000. So it's easier to convert from the millimeter into micrometer by times it by 1,000. So that is 1,000 is our S. I'm going to take our R and times it by S. R is the one in millimeter, and then you times it by 1,000 to get it into micrometer. So T times R times S is our answer. That is going to be D. Next question, 22. Question number 22. This is similar to two of these questions, similar questions we've done before. We're going to follow the same pattern, identify it first, and then match it. 4 students were asked to match the function with the appearance of some cell structures in an animal cells. So you know none of the structures are going to be from plant cells, like the cell wall or the chloroplast is never going to be part of these answers. The functions were listed by a number. mRNA passes through to the ribosome, that is going to be your nucleus, or the nuclear envelope to be more specific. Organizes microtubule to produce spindle during cell division, that is centriole or centrosome, you can say that. Packaging of hydrolytic enzymes that will remain in the cell. So packaging is the key word. Don't just look at hydrolytic enzymes and think of lysosome. It's packaging of that. That packaging is handled by Golgi body. Now, appearances. V is the membrane which surrounds an enclosed inner cavity. This is for SER or RER. Non-membrane bound spherical structures, that is ribosome, is non-membrane bound. It's easier to mess up and confuse between ribosome and lysosome, but there's a difference. A double membrane interspersed with pores, that is the nuclear envelope. So you can be specific, but since this question is not about identifying them correctly, you can even just write it down as nucleus and solve this question. So that is going to be 1 from this. And non-membrane bound cylindrical structure, that is going to be centriole. 2. Membrane bound sacs arranged as a flattened sac. So when you talk about flattened sac, I said earlier that this is Golgi body. So XYZ. Answer is D. The next questions, both are related to cilia and microtubules. Feature of cilia and root hair cells are correct. So cilia, remember the function of cilia is, don't mix up cilia and villi, because microvilli, because cilia is it allows the movement of substances over the cell surface, but the root hair is the one that is going to be increasing surface area. Because otherwise between cilia and microvilli is the one that increases surface area. Allow movement of substances over cell surface. So it's going to be root hairs. Cannot be resolved with the light microscope, so that's going to be cilia. Contains vacuoles. So I'll come to that after this one. More than one present on a cell, so that can be either one. Okay, so it could be cilia or root hair cell, either one. But this is how you're going to figure out your answer. Contains vacuoles. Now remember, vacuoles is only in plants, permanent one. So that's going to be root hair, not cilia. C is your answer. Which are functions of microtubules? Allowing movement of cilia in a bronchus. So remember, cilia is present in our airway and it wafts mucus away from the lungs. So microtubules how is that related to cilia? Because cilia is made up of 9+2 arrangement of microtubules. So yes, it's microtubules that help with this function for cilia. This is correct. Attachment of centromeres during metaphase, that is also with microtubules, because remember in metaphase you have it aligned at the center and you have spindle fibers and microtubules involved. Moving secretory vesicles around the cell, that is going to also play a role here because it forms the cytoskeleton. Remember, cytoskeleton is formed with microtubules, so it's all 3A. Some cell structures are listed: mitochondria, nucleus, chloroplast, and ribosomes. What is the correct order of these cells when listed from largest to smallest? So remember I said earlier that nucleus is the largest, then you'd have the chloroplast, you'd have the mitochondria next, and then the smallest is usually ribosome. So let's use that here. Nucleus is going to be the first one, then you have my chloroplast, mitochondria, and then ribosomes. So it's 2, 3, 1, 4. 2, 3, 1, 4, that is B. Next question, which cell structure? This is the last question. Which cell structure have ribosomal RNA, rRNA? There was another question similar to this earlier. It's going to be all of them. All of them have rRNA. So it's 1A. That's all for this video. I can make a part 2 with more questions for this chapter if you guys want that.